Good morning, everyone. I am Dr. Murugadas, Assistant Professor from Department of Ophthalmology. Today, we are going to see about the anatomy of the angle structures in anterior chamber of eye. So first, let us see what is anterior chamber. Anterior chamber is a space that is a structure present between the cornea and the lens, that is, and the iris. It is bounded anteriorly by the posterior surface of the cornea, posteriorly by the anterior surface of the iris in the periphery, and the pupillary aperture in the center. The content of this anterior chamber is, as we all know, it is an aqueous that has been secreted from the ciliary body or the ciliary epithelium. Okay. So these anterior and posterior borders of that is anterior and posterior boundary of the anterior chamber meet at the periphery of the anterior chamber, that is called as drainage angle of the chamber. This anterior chamber communicates with the extracellular space of iris, ciliary body, and trabecular meshwork. And through the pupillary aperture, it can also communicate with the posterior chamber of the eye, that is behind the iris and anterior to the lens. Okay. Coming to the structures of the anterior chamber, angle of anterior chamber, the structures present are from anterior to posterior. It is first thing is Shalby slab, which is the posterior most border or posterior most edge of the decimus membrane of cornea. And posterior to it is the trabecular meshwork, which is bridged between the ciliary spur and Shalby slab. Posterior to this ciliary is scleral spur, the anterior border of the ciliary body, where its longitudinal fibers gets inserted into this scleral spur. And behind that is the root of iris. So let us see this development of eye as a whole thing. Let us see the parts of the eye that are developed from neural crystal are decimus membrane, that which develops at the 4th month, that is 16th week. Schen's canal that also develops at the 4th month, that is 16th week. Trabecular meshwork at the 5th month, 20th week, and sclera. These are the structures that are developed from the neural crest. In this picture, you can see all those, the the whole part of the maximum part of the eye, that is including the layers of retina, everything has been developed from the neural ectoderm. Okay. Coming to this angle, the cornea, everything is is developed from neural crest. Here you can see this green colored structures are the cornea and angle structures. Then the iris and sclera are developed from the mesoderm. You can see here during the development of eye, it is the optic vesicle that is turned into optic cup by the invagination of this optic vesicle. Okay, this optic cup is going to develop as eye. The blue color, the two double layers seen here are the neuroretinal, the neurosensory retina and the pigment epithelium of the retina. Okay. Progressing to the 32nd week, the angle is at the 32nd week of gestation, the angle is closed by the mesodermal tissues, as we saw in the previous picture. You can see here the mesodermal tissues are occluding the anterior chamber, angle of the anterior chamber. It is the angle is closed by the mesodermal tissue. With 36 to 37 weeks of gestation, the mesodermal tissue starts dissolving and it ends by 38 weeks only, when the angle is completely formed. The clinical significance of this development until the age of development, the gestation of development is the infant born before 38th week of gestation. That is, though it is termed, if the babies are born before the 38th week of the gestation period, they may have incomplete angle and they are at the risk of congenital glaucoma. Okay, let us see the structure, that is corneoscleral limbus. The part where the cornea merges with the sclera, the junction between the sclera and the cornea. This is a transition zone actually, which appears as a translucent. Approximately measures around 1.5 millimeter, okay, in diameter between the clear cornea and the opaque sclera. You can see here the markings in the diagram that this is the posterior limit of the cornea that is being merging with the sclera. And this is the end of the this is considered as the end of the cornea, and this as the start of the sclera. And in between this is called corneoscleral limbus, which measures around 1.5 millimeter in diameter. Centrally, the corneoscleral limb junction is demarcated by a line joining the termination of the Bowman's membrane, Bowman's layer. Here you can see Bowman's layer to the termination of the Decimon's membrane. This line has been drawn by joining the end of the Bowman's membrane and the posterior limit of the Decimon's membrane, which is called as Shalby's line. Peripherally, the sclerolimbal, that is the sclerocorneal limbus, the posterior limit is the junction. The sclerolimbal junction is demarcated by a parallel line passing through the scleral spur. This is called scleral spur, where the posterior part of the trabecular meshwork is getting attached. Here you can see this is the trabecular meshwork which is being bridged between the Shalby's line, that is the posterior limit of the Decimon's membrane, and this anterior part of the scleral spur. The limbus can be divided into three zones. The deep limbus contain the trabecular meshwork, as we saw in the previous picture. From anterior to posterior, it can be divided into three zones. That is, deep zone that contains trabecular meshwork and Schun's canal. This is the Schun's canal, and this is the trabecular meshwork. Then the mid limbus that contains the transitional corneoscleral stroma and intrascleral venous plexus. And see here some plexus, that is, these are the venous plexus that are going to drain the aqueous. The intrascleral venous plexus, which are present in the mid limbus. The superficial limbus consists of, as you know, it is the outermost layer of the eye, the episclera and Tenon's capsule, the conjunctival stroma, and the limbal conjunctival epithelium. The internal scleral sulcus, that is the inner part of this. So this was the limbus what we are seeing from as a total. Now let us see the inner aspect of the limbus region. A circular groove on the inner aspect of the corneoscleral limbus. This part is this corneoscleral limbus. A circular groove present in the inner aspect of the corneoscleral limbus extends from the termination of, that is the posterior limit of the Decimon's membrane, as I said before, to the Shalby's line, to the anterior limit or the anterior most part of the scleral spur. That is, it extends the groove extends from the end of the Decimon's membrane to the start of the scleral spur. This groove is going to accommodate the canal of Schlum externally, okay, and the corneoscleral portion of the trabecular meshwork internally. That is, this part is going to engage the canal of Schlum, and this part is going to engage the corneoscleral portion of the trabecular meshwork. Shalby's line ring, sorry. It is the anterior border ring of the trabecular ring region. Thus, explained by this was demonstrated by Shalby in 1870. The ring marks the transition between the corneal endothelium and trabecular cells. That is, you can see the transition of the corneal endothelium to the trabecular cells is the mark, which is called the ring mark. The ring actually marks the end of the endothelium. That is, beyond the ring, there is no endothelium, corneal endothelium. That is only start of trabecular cells only. It also indicates the termination of decimal membrane. It contains circular arrangement of collagen fibers intermixed with elastic fibers. You can see in the diagram this ring mark actually. This is the region where the endothelium ends and the trabecular cells start. This is called Shalby's ring. Next, coming to scleral spur. It is a wedge-shaped ring, circular ridge. That is the deep aspect of the, you can see here in this picture, the ridge, wedge-shaped ridge, circular ridge. That is the deep aspect of the sclerolimbal junction. This is the sclerolimbal junction, the deeper aspect of which contains the ridge, wedge-shaped circular ridge, that is called scleral spur. It receives the anterior tendon of the longitudinal circular muscle, ciliary muscle insertion on its inner aspect. That we will see in detail about this muscle arrangements in next slides. Okay. Its anteromedial base, that is, this is the anteromedial base, forms the posterior margin of the scleral sulcus and receives the posterior attachment of the corneoscleral meshwork. Hope you can understand that this part is the anteromedial base that forms the posterior margin of the scleral sulcus, and which is going to give the attachment to the posterior part of the corneoscleral meshwork. Here in this picture also you can see that the trabecular meshwork has been divided into uveal, corneoscleral, and juxtacanalicular. Okay. So that is the trabecular meshwork which is giving attachment to the uvea and sclera, uveoscleral, and cornea and sclera, and next to that is a juxtacanalicular meshwork. The scleral spur actually contains collagen and elastic tissue with a circular arrangement, like a trabecular beams of corneoscleral and juxtacanalicular meshwork. The connective tissue compartments are continuous with those of the corneoscleral trabeculae and covered with the trabecular cells. Contraction of the ciliary muscle can exert some pull over the scleral spur posteriorly and opens up the trabecular meshwork. This is a clinically significant point that the contraction of the ciliary muscles can give an pull effect of pull over the scleral spur, which in turn can pull the trabecular meshwork and can enlarge the pores, which is going to be the most likely action of, that is the favoring action of pilocarpine when as it is given as an anti-glaucoma medication. It can cause meiosis and it can have an effect of on ciliary muscle, and contraction of ciliary muscles can pull the scleral spur and opens up the pores of the trabeculae. There are some contractile myofibroblasts like cells oriented circumferentially, oriented circumferentially within the scleral spur, which are rich in smooth muscles, alpha actin and myosin. So this are the, in this picture you can see this trabecular meshes, the three layers of trabecular meshes and the scleral spur that is giving attachment to the trabecular meshes. Again, this picture also explains that these scleral spur cells form tendon-like contact with the elastic fibers of the scleral spur, which are continuous with those of the adjacent trabecular mesh. The changes in the scleral spur cells tone might modulate outflow resistance by altering the trabecular architecture. Now let us see about the trabecular mesh in detail. A sponge of connective tissue. It is that meshwork is like a sponge-like arrangement. A sponge of connective tissue beams arranged as a superimposed perforated sheets. You can see this. The blue, red, and green are the three types of sheets that with a pore size, varying pore size, arranged one over the other. That is superimposed. With the pore size, with the maximum pore size, a uveoscleral junction, uveoscleral measure, and then lesser than this pore size is the corneoscleral, and then next is the juxtacanalicular. The beams are disposed circularly in the chamber anterior chamber angle and extend from Shalby's ring anteriorly to the scleral spur posteriorly in the junction of the iris and ciliary body posteriorly. The inner portion of the trabecular meshwork is referred to as the uveal meshwork, and the outer portion connected to the spur and closer to the Schelm's canal is the corneoscleral meshwork. In this picture again you can see this. This is the inner portion of the trabecular meshwork referred to uveal meshwork, and the outer portion is connecting to the scleral spur and close to the Schelm's canal is corneoscleral meshwork. The line joining the Shalby's ring to the innermost margin of the scleral spur orbitally divided from one another. Between the corneoscleral trabecular meshwork sheet and the endothelial lining of the Schelm's canal is a cell-rich zone. The peri or juxtacanalicular connective tissue zone or endothelial or cribriform meshwork is between the corneoscleral, is this blue colored layer, corneoscleral trabecular meshwork sheet and the endothelial lining of the Schelm's canal. This canal, this yellow colored area is the called juxtacanalicular zone. The extracellular space of the trabecular meshwork contains hydrophilic glycosaminoglycan and collagenase material that influence the resistance to aqueous flow by the meshwork space. Coming to the uveal meshwork. The inner uveal meshwork, the inner most of the trabecular meshwork, has 1 to 2 layers, is made up of a cord-like trabeculae that taper anteriorly. The innermost trabeculae pass from the ciliary muscle to the region of Shalby's ring. This picture again you can see it is a 1 to 2 layers. It is with the EVL measure is made of 1 to 2 layers. A cord like trabeculae that tapers anteriorly and but means increases in thickness by the posteriorly. The innermost trabeculae passes from the ciliary muscle to the region of Shalby's ring. From ciliary muscle to the Shalby's. Posteriorly, there are 2 to 5 layers. The outer layer have a more circular oriented, more circular orientation concentric with the limbus, resembling that of corneoscleral sheet. The trabeculae may be connected either with the circular or radial muscle fibers of the ciliary body or within the meridional ciliary muscles. The measure can be attached to the ciliary muscles, either the circular or radial muscle fibers. Okay. Anteriorly, the uveal trabeculae converge to end by joining the periphery of the decimus membrane. As we already told, decimus membrane, the periphery of the decimus membrane, the inner part of the Shalby's ring, where the corneal lamellae or the corneal sclerotrabeculae. The cellular lining of the uveal trabeculae is contiguous anteriorly with the keratocytes in the deeper region of the cornea and its endothelium. The individual cords of the uveal trabeculae have a diameter of 4 to 6 micrometer in the mid region, being thicker posteriorly and narrower anteriorly. The intertrabecular spaces range in size from 20 to 75 mm. Coming to corneoscleral measure, it is made up of flattened perforated sheets, about 5 to 12 in thickness, picometer in thickness in the mid portion and separated from each other by 5 to 20 picometer. Anastomosing band between superficial and deep layers ranging from 2 to 3 micrometer in thickness delineate the intertrabecular space. That is, the anastomosing band between the superficial and deep layers range from 2 to 5 micrometer in thickness, which delineate the intertrabecular space. There are approximately 8 to 10 trabecular layers with a total width of 120 to 150 micrometer. Anteriorly, the sheets converge, obliterate the intertrabecular space, and merge with the inner corneal lamellae, and the trabecular cells interface with the keratocyte. Posteriorly, they are inserted into the scleral roll. The intratrabecular space of the outer layer of the corneoscleral sheet vary between 2 and 20 micrometer and are therefore narrower than the uveoscleral mesh. That is, the pore size of the corneoscleral measure is less than the uveoscleral measure. The space decreases in size from within to outward. Trabecular structure. The basic structure of the uveal and the corneoscleral beams are as follows. That is, each sheet has a covering of trabecular cells, a subcellular cortex, and an inner collagenous core exhibiting randomly distributed elastic tissue. The collagen is oriented in the long axis of the trabecular beam. The trabecular cells are elongated in the long axis of the trabecular sheet, 4 to 8 micrometer in thickness centrally and about 120 micrometer in length. The nuclear bulge is frequently present and occasionally a cilium is present. The neighboring cells make contact with the long cytoplasmic process and opposed surface are attached by maculae, adherent, and gap junctions. This picture demonstrates the trabecular mesh cells and the extracellular matrix in the UV scleral beam. And this is the corneoscleral mesh granule. You can see the collagen fibers and elastic fibers. The cells show the usual organelles, including the central nucleus, moderate amount of mitochondria, rough and smooth endoplasmic reticulum, well-developed Golgi apparatus, lysosomes, pinocytotic vesicles, and vesicular bodies. The cells are actively phagocytic, involved in a self-cleansing mechanism, which keeps the trabecular filter clean. Let's come. Let us see the peri or juxtacanalicular connective tissues. The connective tissue juxtacanalicular zone. The peri or juxtacanalicular connective tissue zone invests Schell's canal in its entire extent. On the trabecular aspect, it has been termed as a endothelial mesh. You can see in the trabecular end, it has only endothelium. So it has been represented as endothelial mesh in the trabecular aspect. It has a thickness of 2 to 20 micrometer and is interposed between the endothelial lining of the canal and the outermost corneoscleral trabecular sheet. The region consists of 2 to 5 layers of loosely arranged cells embedded in the extracellular matrix. The cells exhibit long, slender process and attach to one another irregularly. You can see in the picture that they are attached to one another irregularly, arranged irregularly by maculae occludens, desmosome, and gap junction. The space exists between these cells up to 10 micrometer in width, through which aqueous humors can percolate to reach the endothelium cells of the Schell's canal. The pericanalicular cells have important phagocytic and secretory properties related to the self-cleansing role of the mesh and to produce the production of extracellular matrix respectively. This portion of the drainage system is thought to make a major contribution to the outflow resistance, not only because the pathways are narrow and tortuous, but because of the presence of extracellular proteoglycans and glycoproteins. This is a 360-degree view of this angle structure, that is Schell's canal. The narrow circular tube around 36 nanometer in circumference, lined by the endothelium. It lies in the outer portion of the inner scleral sulcus. It conducts the aqueous humor from the trabecular region. It conducts the aqueous humor from the trabecular region to the episcleral venous network via collector channel. Here you can see this. This is a Schell's canal. The inner collector channel, and this is the external collector channel. Schell's canal, a zone of pericanalicular connective tissue, separates the inner and outer wall of the Schell's canal from trabecular mesh and sclera, respectively. The lumen is actually elongated and oval, or sometimes triangular in cross section. You can see here the Schell's canal, the cross section of the Schell's canal. The endothelial lining, there was a controversy over whether there was an open communication between the anterior chamber and the canal of Schell's or not. It is now generally considered that the vesicular configuration of the endothelial can provide a mechanism for direct communication between the extracellular space of the trabecular zone and the Schell's canal. Schell's canal is lined by a single layer of spindle-shaped endothelial cells oriented parallel to its circumference. In the inner wall, they are 40 to 120 micrometer long. 4 to 12 micrometer wide and 0.2 micrometer thick. They are connected by poorly defined tight junction and macroadherent and by occasional gap junction with cells of pericanalicular tissue. The luminal surface of the cells exhibit sparse microvilli. Their cytoplasm shows numerous free ribosomes and microfilaments and pinocytic vesicles. The most prominent feature of the inner wall of the canal of French canal are the giant vacuoles. These are the imaginations globular or meridional section. The vacuole globular on meridional sections. The vacuole measures around 4 to 5 nanometer in width and 25 nanometer micrometer in length and usually are oriented along the long axis of the cell. A smaller proportion of the vacuole also communicate with the canal of Schwann via a luminal opening or pore, by which a transcellular channel may be formed. These giant vacuoles, the studies have demonstrated the formation of endothelial vacuole to be a pressure dependent, so that the inner number and the size are reduced at low pressure and increased at high pressure. The endothelial cells of the inner wall of the Schwann canal have the ability to transfer the aqueous in bulk by cyclic pressure dependent process involving the formation of vacuole and transcellular channels. It is accepted that the aqueous enters the canal of Schwann almost entirely by transcellular route, and only about 1% of the total conductance of the inner wall of the canal can be attributed to the non-vacuolar channel found in the narrow marginal portion of the endothelial cells. Coming to outer wall, the endothelial cells of the outer wall of the Schwann canal are longer, flatter, and smoother in outline than those of the trabecular side. The cells are joined by zonula occludens, shows a rare giant vacuole on their luminal aspect. Rest of the basal lamina, which is thicker and more continuous than that of the trabecular cells. Finally, the collector channels. The collector channels arise at the irregular interval from the outer wall of the Schwann canal. The collector channels are lined by vascular endothelium similar to that of the outer wall of the Schwann canal. They are 25 to 35 in number and drain into three interconnecting venous plexus: the deep, mid-scleral, and mid-scleral and episcleral venous plexus. Up to 8 vessels drain directly into the episcleral venous plexus are known as aqueous vein. No valves are present in this system. So this is the total. It is a structure of angle. So far we have seen and how the aqueous has been drained through this angle structure. Thank you.